माझं तर किती वेळा पडली.
ते पण सेंद्रिय लागले.
उसको ये करके रखते ओके ये रहने दो
उसके पहले
मैडम वांट्स टू जस्ट चेक
the slide is not moving.
उसका भी तो रिमोट नहीं है क्या है ना फिर उससे दे उससे करेंगे मैसेज चेक ना तो आसेल मैडम इसे सब
First or should I pick that first?
which one
No, this first I'm starting with this.
yeah okay एक मिनट
क्लियर है ना तू स्लाइड शेयर क्यों करेगा
ये थोड़ी देर में इसको बिठाता हूँ
आप स्टॉप करो मैडम
नहीं रहने दो मैडम
नहीं उसको स्टार्टिंग का कुछ शेयर करना है
अच्छा ओके
एक मिनट
ठीक है किसको डीपी को
ठीक है किया
मैडम
इसे शेयर कर दिया
हमारा देखते हैं मैडम कैप्टन मैडम ही इज कैप्टन प्रवीण नायक चलेगा बैकग्राउंड बहुत सारा कुछ दिख रहा है हाँ आया ना उसने बनाया ना टीपी ने he wants to share that Himanshu sir can we start
Yes, madam. Yeah. So good morning, one and all.
good morning
Yes, respected Dr. Shubhada Chiplunkar, madam, former director of ATRAC, Advanced Center for Treatment, Research and Education in Cancer, Tata Memorial Center, former principal of RJ College and now the director of academics at Hindi Vidya Prachar Samiti, Dr. Usha Mukundan, madam, members of Women Development Cell of RJ College, also my teacher colleagues from different departments, and dear students. It gives me immense pleasure in inviting you all for this Savitribai Phule lecture series being organized by WDC of RJ College. Friends, it's a pleasant morning indeed. With me, all of you are very soon going to witness meeting of two academicians, two scientists, two mentors, two role models, two friends, and above all, two wonderful human beings. In this meeting today, and the occasion, which is a unique of its kind, you don't find such functions in the usual function book of any education institute. To give you a little background, in 2016, our principal, Dr. Usha Mukundan Madam, received Savitribai Phule Adarsha Shikshika Puraskar from University of Mumbai. That was the moment when WDC decided that this achievement shall go in the chronicles of RJ College as a felicitation of strength and perseverance of a women. To mark this occasion, the birth date of Savitribai Phule, 3rd January, was a date selected wherein we shall felicitate a woman achiever who has made a mark in the field she chose and has climbed the ladder to the level that serves as a role model to others, giving a very silent and subtle message. Girls too can dream, girls too can achieve, and education is for them too. In this series today, ladies and gentlemen, we have Chiplunkar Madam with us. I fall short of words when I have to speak about Madam for the simple reason that the great support that she has always extended for fulfillment of the research aspirations of her students can't be expressed in words. She has been instrumental in having many of her biotechnology students and pure science students completing their small research projects and research dissertations at her center. And apart from providing a working desk to them, she also has been their mentor for that period they spend on her center, explaining them what time management is all about in research, how to shed the habit of ignoring small findings but putting them in place, meticulous designing of the experiments, and perfect interpretation of data. Dear friends, let me give you a gentle reminder in the recently completed annual academic prize distribution function, wherein we have felicitation of one of our alumni, Dr. Vaibhav Chikane, who is currently doing his postdoc in Denmark, had started his small research stint only in Madam's laboratory. The passion with which he speaks about Usha Madam, with the same passion he speaks about the Chiplunkar Madam, that goes to say that our guest today has been such a wonderful researcher and above all such a wonderful mentor and human. There are many such careers that she has shaped, and I take full advantage of this opportunity today that on behalf of all of us, I express a sense of gratitude and thanks to Madam who has come on our campus today. And from the very conference room of our own, she's going to deliver her talk. Madam, thank you very much being with us today morning, giving your valuable time here. I welcome Madam and everyone else. I request my course IQSC coordinator and colleague in the office, Vice Principal, Dr. Seema Ratnaparki Madam, to give a formal introduction of the Madam. And that shall be followed by words from Usha Madam and a moment of felicitation of our guest today on this 5th Savitribai Phule lecture series. Dr. Seema Ratnaparki Madam.
Thank you, sir. Thank you, Himanshu, sir. Good morning, everyone. I welcome once again Dr. Shubhada Chiplunkar. It gives me an immense pleasure to introduce our today's chief guest, Dr. Shubhada Chiplunkar. Dr. Shubhada Chiplunkar has been the former director of Advanced Center for Treatment, Research and Education in Cancer, that is ATRAC, Tata Memorial Center, Navi Mumbai, and also head of the Cancer Immunology. She obtained PhD from University of Mumbai in 1983 and postdoctoral fellowship from Max Planck Institute, Germany in 1984 to 1986. She has 35 years of research experience in the field of cancer biology and immunology. Her research interests are understanding immune dysfunction and cancer patients and development of immunotherapeutic strategies for cancer using gamma delta cells. She has 123 publications and 2 patents on her name. Madam has received many awards. I'll just name few of them. Senior Scientist Oration Award by Indian Immunology Society in 2007. Presidential Oration by ICAR 33 in 2014. Professor Milton Yotwin Oration in 2017. Presidential Oration Immunology Society India Award in 2019. There are many awards she has received. She has been appointed as an office bearer of more than 20 national and international funding agencies and task force committees. Just to name few, she is a task force committee member of DBT, BIRC, CSIR, ICMR, Senate member of IIT, Academic Council of Universities. She is a president of Immuno-Oncology Society of India and Mumbai Immunology Group. At present, she is a member of the Education Committee of International Union Immunological Societies. So I think this is, if I read her CV, then I think 1 hour will be less. So as Himanshu sir said, she's an icon, she's an idol for us. So Madam, once again, I welcome you for this today's program. And I request Usha Madam to say a few words and felicitate our today's chief guest, Dr. Shubhada Chiplunkar. Over to Usha Madam. Thank you, Madam.
Thank you very much, respected Principal Dr. Himanshu Davda, IQAC Coordinator Dr. Seema Ratnapaki, Vice Principal Captain Praveen Nayak, and my very dear friend. We are very, very proud of you, Dr. Shubhada Chiplunkar. At the outset, we are also thankful to you for accepting our invitation and for being with us today morning to deliver this lecture on the occasion of the first teacher of this country, Savitribai Phule. I have known Chiplunkar Madam for about 2 decades. She comes out to be a very, very gentle person, very humane, but at the same time, I would say an iron lady when it comes to selection of research scholars, selections of colleagues in the center, and the research work. She does not compromise on that. And I think that is the reason the immune system of the country is strong. So today in the pandemic and coronavirus has given us a new mantra. So anything in the name of immunity is savable. But Dr. Chiplunkar does not believe in that. She believes that science requires experimental proof. It should be validated and then only it should reach the public. Chiplunkar Madam is also a good example as to how you should slowly and steadily rise to the pinnacle of any institution. At the same time, when you reach the heights, you also remember from where you have come. And this helps in leadership because you know how to handle your colleagues, how to take very, very important decisions without compromising on anything. And today's lecture, I think she's the right person. And we are very fortunate to have with us today morning, Dr. Chiplunkar. On this occasion, I'm also thankful to my own student and current principal, Dr. Himanshu Davda, because he floated this idea to start the lecture series as a tribute to his teacher, that is myself. And I think for any teacher, there can't be a greater occasion when your students look at you very lovingly and they are always with you in your journey. So thank you very much for that. And Chiplunkar Madam, we would like to felicitate you in our own traditional way. So once again, thanks a lot.
Thank you,
Yeah,
madam, and
please
I
join
request
us.
you to
Yeah.
do the honors.
Thank you. Let us see.
Yes. Hemant sir,
Camera.
camera.
थोड़ा सा वर्क करने का
Yeah.
थोड़ा सा करो और करो. So Ganpati Bappa Moria.
Yes.
Thank you so much. And especially handmade, so we want to also take care of the local community. Thank you.
Thank you. Thank you.
Yeah, मैडम के ऊपर फोकस कर दे. नहीं पहले कैमरा मैडम के ऊपर रख. कैमरा मैडम के बेस पे लगाया.
This is not open.
मैडम देखिए
क्योंकि मैंने ऊपर किया था कैमरा. दिख रही है कुछ.
हाँ दिख रहे हैं.
So good morning. I'd like to thank the principal, Dr. Himanshu, Vice Principal and all staff of Chiplunkar College. And a special thanks to my friend and colleague, Dr. Usha Mukundan. I feel deeply honored and humbled that you have asked me to give this talk on the occasion of the birth anniversary of Savitribai. My association with Dr. Mukundan dates back to several years. We both were representing the university on several committees, and we have shared our experiences both in academics and in personal life. I found Usha to be a wonderful person, a wonderful human being, and a great teacher. Whenever a call comes from Usha to me to take someone as a trainee in my lab, I've always accepted it. And I must say that these have turned out to be an asset to the lab and wonderful students. And as you mentioned, by Chikring, who worked in my lab, and others also. So I feel it's a tradition of the Chiplunkar College to nurture excellent talent. So thank you once again, principal and staff of Chiplunkar College, for honoring me. At the onset today, I would first pay tribute. Pay tribute to I would pay tribute to Savitribai Phule, who was a pioneer of the Indian women's education, a wonderful, outstanding educationist who worked for the upliftment of women, empowerment of women, and started the first education program in India. What I'm going to do today is talk about my journey into cancer research, and in the process, how it has been a learning and educative process for me to a self-discovery, I would say. I started my career after my graduation in microbiology from KJ Somaya College, and we were the first 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 team of graduate students graduating BSc, from the college. There were hardly any colleges offering MSc programs, and I came to know that at the Cancer Research Institute, they were offering MSc, and I applied for it. I joined Cancer Research Institute, and some of you may be remembering this old building, which was in Parel, next to the Tata Memorial Hospital. I stepped into this center in 1975 through a very stringent process of interview, which for a young graduate was something very, very different. I got selected, and it was with Dr. Nandini Sheth that I did my MSc, and it exposed me to cancer research. It was a stepping stone into cancer research. I completed my MSc and in 1979, and I was determined, of course, to do my PhD here. Again, had to go through a selection process. And I must share with you that I got married in 1979, and my husband, Chiplunkar, is an engineer, and the Chiplunkar family gave me full support to proceed and go ahead with PhD. So I enrolled for PhD in 79 with Dr. Kumud Karande. So this is the Cancer Research Institute, and we moved to this ATRAC, which was mentioned earlier as the Advanced Center for Treatment, Research, Education in Cancer. The new center, we moved here in Navi Mumbai in 2002. This is the Tata Memorial Hospital, and the new building, the tall high-rise which you see here, that was where the Cancer Research Institute was. So now we have moved to the new campus. I did my PhD here in the old CRI. And Dr. Kumud Karande was my mentor. A wonderful personality. She was as she looks. Very smiling, always encouraging. And I worked with her in understanding the role of murine mammary tumor virus in breast cancer. So basically, we were doing, because of my MSc experience in radioimmunological assays, we started designing assays for detection of MMTB antigens and autoantibodies. But one thing I must share with you all, because since this lecture is meant for the students, I would be sharing you my experiences, the euphoria at times, and how your euphoria in research doesn't stay long and brings you to ground level very soon. So when I joined for my PhD program, Dr. Karande was not here. She was abroad. She was in the Netherlands. And I was told that she'll be coming back after a year. So your registration will happen after a year. Now, this is quite a depressing thing for a young PhD student to be told that your guide is not here. You'll get registered after 1 year. But you start working on the project. So, and those days, the letters would reach after 8 to 12 days. So she was exchanging letters with me, and we had already decided on what I should work on. And I started standardizing things on my own. Fortunately, things worked. And before she came back, I already had the assay in place. So she was very happy about that. That we had already standardized the process for labeling viral antigens and doing immune precipitation assays. I completed my thesis work, and unfortunately, Dr. Karande had an accident, and she passed away in that accident, so she was not there for my viva voce examination. Another shock in life. And that is when Dr. Langal, who was heading the department, came and said that I'll be there for your viva and it will go smooth, don't worry. The viva went off, and in 1983 I got my PhD. And Dr. Langal was very keen on taking me as a scientific officer in the Cancer Research Institute in her group. Again, I had to go through the process of interview, and I got selected and was designated Scientific Officer B. With dreams in my eyes and and enthusiasm to do research, I stepped into the immunology department. And in those days, there was a big hype about hybridoma. Dr. Karande, my boss, already treated hybridoma,
ঘরে তো নকুল ভাঙানো
We were able to initiate the work here. And Dr. Kanwal took over the reins. And she said that we will start doing the hybridoma work here in the lab. And it was that time when George Kohler had, and Caesar Milestein, whom you see in the picture there, had made hybrids. They were actually working, and they were not making hybridomas, they were making somatic cell hybrids. And landed on this. So this was serendipity that they landed into making hybrid clones to specific antigens. We standardized this technique, and mind you, it's a very, very laborious work. Those days it was extremely laborious to make murine monoclonal antibodies. We selected targets like the alpha-fetoprotein, which is expressed in the cancer cells, liver cancer cells. We made monoclonals against oral cancer antigens. We made against leukemic antigens. We also went ahead and made anti-idiotypic antibodies, which is even more complex. It was fun, it was back-breaking work. You are dealing with 600, 700 clones, screening them, freezing them, thawing them, testing them, characterizing them. Oh my God, it was a factory which we were doing. But I enjoyed every moment. I enjoyed working and doing these clones. It was then that there was a visitor in the institute from Germany, Professor Stefan Kaufmann. He came to our lab, and he was looking for postdoctoral fellows. I had a discussion, Dr. Ganguly introduced me to him, and I remember I traveled with him in the taxi from Parel to the Taj Hotel, and he interviewed me in the taxi. And we kept discussing about the work, and at the end he said, when we got down from the taxi, he said, I offer you a postdoc position in the lab. So, you know, any person would have got excited, and I was, of course, I wouldn't deny that, but I asked him, I have not given you my decision. So he laughed and he said, why, don't you want to come? So I said, no, I want to know how big is your lab? How many students you have? And he started laughing, and he answered all my questions, he answered all my queries. And I joined Stefan at the Max Planck Institute of Immunology in Freiburg, a very beautiful place. I would say it's one of the best places in Germany, the Black Forest. That's called the Black Forest. So I joined FDI, and Stefan gave me the project of making cytotoxic T cells or lymphocytes against M. leprae. And I said, well, now nice. I will be working on microbacterial immunology and learning a lot. And I must share with you again another experience. He was introducing me to different lab colleagues. And one of the colleagues, she said, hey, you're here to do M. leprae work? I said, yes. She said, pack your bag and go home. I said, what? This was not something I expected on my, on the first day. She said, I worked on it for 1 year and I've given it up. So I started working on another area. So I said, what the hell? I've left my family, I was married, I left my family, I've come here to hear this, to somebody who tried and gave up the project, and now this project is with me. So I went to Stefan and said, Stefan, I want to have a word with you. And imagine this was happening on the first day of my postdoc. And Stefan said, what is it? Why are you so upset? I told him everything, and he said, Shubha, each person, all are not the same. Maybe you will be successful. You have to work hard for it. So I was not very convinced, but I took up the challenge. And I tell you, it was a lot of hard work, because M. leprae is not at all a good bug against which you can make T-cell responses. So I tried hard. I used to work with mice, immunize lots of mice, take out lymph node spleens, make T-cells, grow them. And T-cell cloning was a new thing in those days. Clone them, do limiting dilutions, clone them, do flow cytometry, characterize them. And I would never get antigen-reactive T-cells. But I didn't give up. And Stefan, he was dynamic, exuberant, and a voracious reader. In those days, we used to sit in the laminar floor and work. He used to come from behind and he would always say, continue working, don't look back, I'm reading out a paper to you, keep your ears open. This was the way he dealt with us, he worked with us. But I enjoyed that too. I enjoyed his pressure, the way he got work done for us. Why I enjoyed it? Because I got full freedom to do what I want. After 1 year, I made a drastic change in my approach. When I were given subcutaneous injections, I read through papers and I said, no, I'm going to try intradermal. He said, what difference will it make? Do you think it will make a difference? I said, let's try, because a paper published in 1941 by Shefford has used intradermal injections, and they've got some good responses. And we had immunized a whole bunch of animals. And we opened the first animal and we saw pea-sized lymph nodes. And I got Stefan, and typical Stefan, he said, oh, that's an infected animal. So I opened another one, and that was again a very large lymph node. And he said, Shubha, you're in business now. That's all he said, and he went away. And we then started making clones. We made cytotoxic T clones. And yeah, I must share with you that this, my frustration, I have also written in the book, Leelavati's Daughters, which was Indian National Science Academy. I had invited some of us to share their experiences, and I had written particularly in the statement, so how frustrating it was to make these clones. But finally we got a very nice papers, and simultaneously, it was always, I don't know, I've always been doing that. Cytotoxic T lymphocytes were not happening, so I told Stefan I want to work on something else. He said, what is it you want to work on? And that time, recombinant cytokines were coming in, people were making recombinant cytokines. And Charles Dinararo had made recombinant interleukin-1 and sent to Stefan. And Stefan asked, who wants to play with this molecule? And I said, I would like to play with it. And so simultaneously, I was working on B-cell growth and differentiation by interleukin-1. So imagine two projects going on simultaneously, working day and night in the lab. And finally, both worked and both gave us very good results. Now, I must also pay my respect to one of my, in whose house I stay, and she's from Mashwads. This is not a picture taken then, it was just 2 years ago when I visited her. I'm still in touch with her. She and Gustav Schwartz, they gave me a home away from home. Wonderful people. They'd seen an Indian for the first time. I stayed with them, and I had a wonderful stay with both of them. You need some people around you like that when you're away from your own house. And I was fortunate to have Ilma and Gustav with me. What's happening? Yeah. After coming back from Germany, I wanted to go back to India after 3 years, and I told that to Dr. Kaufman, and he said, you can stay here if you want, but I had made up my mind and I decided to return to India. It was at Cancer Research Institute, things had changed. Dr. M.G. Dev was the director of the center, and he had a big program going on the anti-leprosy vaccine. He was doing a big trial on the ICRC anti-leprosy vaccine. This was an isolate made at the Cancer Research Institute, and they were spearheading the vaccine trial. So there were over 30,000 household contacts of leprosy patients who were getting vaccinated with this vaccine. And he was looking for someone to take care of the laboratory research on ICRC. And my CV and experience in mycobacterial immunology fitted well. Again, went through the entire process, interview process, and got selected. And when I told this to Stefan, Stefan said, let's have a collaborative project and continue with the collaborative project. So we wrote an Indo-EU project, Indo-EC, that I wrote in commission, and we got a big grant to this work on immunology and immunoprophylaxis of leprosy. Of course, it was Dr. Dev and his guidance that we could do 2 big projects under his leadership. It was a big learning experience because then it was not just focused with bench work or lab research. I went to the vaccine field trial areas, visited them in Sholapur, Rathur, Usmanabad. I could see, this is the first time I came in close contact with leprosy patients. And my working area was in Meerut, in the Richardson Leprosy Hospital, where we were dealing with multi-drug resistant cases. This changed my whole perspective, and I realized that we cannot remain in our own cocoon doing lab work. There's so much outside, there's so much suffering outside. And what's the use if your research cannot get translated to the patients who need them? My visit to Richardson Leprosy Hospital was almost once in 15 days to bring samples. And so I interacted with the mission hospital. And I was amazed at the dedication with how the team worked there. Anyway, we decided not to characterize the ICRC bacillus because there was no evidence about whether it shares antigens with M. library. So we did, and we characterized them, we made antibodies, we looked at reactivity of seras, we looked at reactivity of monoclonals, and showed that there were common antigens. We also looked at T-cell responses, whether they generated in vaccinated patients before or after vaccination. Mind you, these are multi-drug resistant cases. So it was really a challenging thing to get a T-cell response in MDT resistant cases. We looked at natural killer cells, we looked at lymphokine active, so we left no stone unturned to fully characterize the immune responses, the immune scenario to ICRC. While that was going on, I always kept telling to myself that, look, you're working in a cancer research institute, you're working with leprosy. Tomorrow somebody may come up and ask you, what have you done in cancer? And what would you answer then? So, working in mycobacterial immunology in Stefan's lab had also given me some idea about a very unique subtype which was coming up. And that was Susumu Tonegawa had isolated genes for the gamma delta T cells. Their function was not known. Nothing was known about these cells. But always at the back of my mind were gamma delta T cells. And I thought that when I go back to India, I would perhaps work with these cells. And now you all know that cancer immunotherapy is at the forefront of cancer research. And that was this, I'm very fortunate that I got such a field and I really enjoy working in cancer immunology. So I started working, then joined, of course, after this as an officer in charge, it was called, in cellular immunology in '94, then became head of cancer immunology in 2001. And I had a very defined focus in front of me. I said, I will work with understanding the reasons for immune dysfunction in cancer patients, how the tumor microenvironment works, how it is influenced, or how it influences the immune system, understand the role of innate immune cells in cancer, and that meant the gamma delta T cells, and also study the immunosuppressive networks, and to look at all other cell types, the immunosuppressive cell types, and their crosstalk with gamma delta T cells, and the final aim would be immunotherapy of cancer. Just to share with you, and in between I'm going to just share with you some of the work which we did in this area. As you look, the tumor, it's a very complex microenvironment. You have a tumor bed, then the tumor stroma, and you have a lot of cells infiltrating into the tumor bed. And these are immune cells, like T cells and B cells. And with that, you have a whole lot of immune suppressive cells, like the MDSCs, the regulatory T cells. So it's a tussle between the two, the good and the bad. And when I teach my students, I always say, you know, they're like two sumo wrestlers facing each other, throwing challenges at each other. That is the tumor on one side and the immune system on the other. The tumor is far smarter. It has evolved all ways of avoiding being recognized by the immune system. Like immune system you put up one challenge, but tumor has already counteracted that challenge. I'm not going deep into the understanding, but you know, a lecture can be given on how tumors evade immune recognition. And that is the challenge, to have a good immunotherapeutic approach against such a strong tumor cell, which can evade being recognized. So coming to the unexplored world, I would say, of gamma delta T cells. I started and I decided that I will work on gamma delta T cells. Okay, then you need probes, you need antibodies. And you'll be surprised, those days there was nothing available. Go to the catalog, there was not a single antibody to gamma delta T cells. And I did not mention to anyone that I want to work on gamma delta. People would have laughed at me that what are you going to work on when you have no reagents. And that was when my student, Anu, came to the lab. And I told Anu, you're going to work on gamma delta T cells. She was not, because it was not a very popular cell type those days, you know. So I knew it would be very frustrating for her if I put her directly on that. So I told her to work with cytotoxic T cells and NK cells and to look at their frequency. So she was busy with doing her work, while I was busy looking for how to get reagents. And then I looked to one of the scientists, Dr. Dieter Kablitz, who's again in Germany, and who had just met once. And I came to know that Dieter has made monoclonals to gamma delta T cells. I wrote to him and he said to me, oh, Shubha, you're late. I'm just licensing it to one of the companies. I said, Dieter, can you give me? And in those days, getting reagents, and I'm talking way back in the 80s, it was very, very, very difficult. So Dieter said, okay, I'll see what I can do. And he sent me a gift of subset-specific gamma delta antibodies. And that, then there was no turning back. We started working on this unique cell type. And I'll just tell you briefly, it's a T cell which behaves like a B cell. It doesn't require antigen processing and presentation like a classical T cell. It can look at antigens directly. And the very common subtype is the V gamma 9, V delta 2 subtype. So the gamma and the delta chain come together like the alpha and the beta chain to form the TCR. This is the gamma 9 delta 2. And they're highly cytotoxic. They can kill tumor cells, they can kill infected cells. And because there's no antigen presentation required, they can work across MHC barriers. Now, one of the compounds which activates gamma deltas are the bisphosphonates. Now, bisphosphonates are given to patients to prevent osteoporosis or to prevent bone metastasis, that is spread of cancer to the bone. It's just routinely given. So these bisphosphonates are known to activate gamma deltas. Of course, there is not much information on how they activate, except that they inhibit the enzyme farnesyl pyrophosphate synthase. With this clue in hand, we started working, because if you inhibit the downstream farnesyl pyrophosphate synthase in the mevalonate pathway, I forgot to mention, in the mevalonate pathway, there is upregulation of the upstream isopentanine pyrophosphate. And this isopentanate pyrophosphate is recognized by gamma delta T cells. Now in cancer cells, mevalonate pathway is dysregulated. So there are tons of IPP which is produced internally, and therefore gamma deltas can get activated and recognize cancer cells. So this was the first paper we published in '99, and we were so excited because here we showed that another antigen, that's a heat shock protein, HSP16, is recognized by gamma delta T cells at antigen. So we were in business. We said we look at all cancers now and ask all these questions. So we looked at oral cancer, lung cancer, breast cancer, cervix, gallbladder, pancreatic, colon, leukemias, acute lymphoblastic leukemias, bone marrow transplantation. And this is the work done by my lab over the years to understand what ligands these gamma deltas recognize on the tumors, what is the clonality of these gamma deltas in tumor versus peripheral blood, because there is a sequestration, of this subset. How do you improve anti-tumor cytotoxicity? How these gamma delta T cells migrate? What are the chemokines and cytokines that dictate their migration from the peripheral blood to the tumor? What are other ligands? One of the interesting ones is the alkylamines, which is found in black tea. And I'll tell you later how we expanded on that. Then toll-like receptors, what regulates functions of gamma delta T cells, and we landed up with Notch. We looked at a subtype of gamma deltas, which is a pro-tumor subtype, which is the gamma delta 17. We looked at crosstalk of gamma delta T cells with immune suppressive cells. We also looked at crosstalk of gamma deltas with bone cells, those are the osteoclasts, because bisphosphonates are given to prevent osteoporosis. They prevent the action of the osteoclast. We started looking on epigenetic regulation of gamma deltas. We are looking at metabolic pathways in gamma deltas. And we did a lot of collaborations with IIT Mumbai to use nanoparticles of zoledronate, to use scaffolds, and to see how these can be used to activate, or how can you deliver zoledronate nanoparticles, or how can you use T cell scaffolds to harbor T cells. And finally, we are also looking at how hypoxic tumor microenvironments. So I feel almost every question over the years we have addressed, or trying to address in gamma delta T cells. I always kept my focus on three aspects: basic research component, a translational component, and finally, which can go towards a clinical trial. See, you may plan that, but it may not happen like that. You are not sure about what would basic research question is. So you have to be sure in picking your questions. What basic questions are you asking? And what is the translational component coming out? So some of the work which we showed that CD3 zeta chain is a good biomarker in oral cancer. We identified a subgroup in leukemia patients which showed good prognosis based on the TCR clonality. We looked at urinary cytokines as prognostic biomarkers in bladder cancer. We identified delta 17 as a pro-tumorigenic subtype in gallbladder cancer. So that means if you block these the delta 17, you get improved prognosis, and then finally the role of bisphosphonates. And then this is how we participated in several trials which were initiated by our clinicians, trying to answer the basic research questions in their clinical question. So coming to gamma delta T cell cytotoxicity, now this was way back in 2010, we showed that if you take tumor cells and treat them with bisphosphonates, as I mentioned to you earlier, IPP will get upregulated and gamma deltas will fiercely kill these tumor cells. Just an overnight incubation of tumor cells with zoledronate can make them susceptible to gamma delta T cell killing. And we looked at the mechanisms, which are the molecules involved, the NKG2D expressed on gamma deltas and the MIK expressed on the tumor cells. So the entire pathway we could trace. This is actually a good video, but I'm not showing them due to lack of time. But you can see how beautifully gamma deltas kill. Very recently, just a few months ago, we have now a method, LCMS-based method, to measure the intracellular metabolite IPP in tumor cells after zoledronate, and we were very successful in standardizing this technique. And this was under the DBT Unit of Excellence project, one of the objectives. And so we have this technology. So this will be a wonderful handle for the clinicians to see which patients respond to bisphosphonates, and that would be very important in treating skeletal malignancies. This again as a part of the DBT UOE project. With Dr. N.T. Banerjee's group, she made this CZ nanoparticles for zoledronate, and we showed that they could beautifully expand gamma delta T cells. You take peripheral blood mononuclear cell, throw in the nanoparticles, give you a particular protocol of expansion, and then you'll see there's an outgrowth of gamma delta T cells. HD map, which you see in the figure here, is it's the analog of IPP, which is known to act as like a standard antigen which activates gamma delta. We are working on this. We have shown that if you treat cells with CZ, you get a complete lysis of tumor cells. This is just image analysis done with the tumor cells alive. So the study still continues. Then, so the aim was to provide a proof of evidence to show that gamma delta T cells are killer cells. Okay, we showed that they kill tumor cells. Another important aspect, whether they mediate ADCC, antibody-dependent cellular cytotoxicity, whether they are good in vivo in imaging the tumors. So we did a SPECT by using radiolabeled gamma deltas and looking for their imaging in vivo in immunodeficient mice. So establish tumor xenografts, take surgically excised tumors, make xenografts, and then inject the antibody which we had made in the lab against the oral squamous cell carcinoma, and so show that it targets. Then we use the idea of taking this monoclonal, which is able to recognize the tumor and hook it on to gamma delta T cells via the Fc receptor. Here is a cartoon which explains that. This is the gamma delta T cells. Utilize the Fc receptor on the gamma delta T cells, hook in the antibody with its Fc quotient, and the FAB will target the tumor, and show that gamma deltas can kill tumor cells by ADCC mechanism. As I said, I'm not going into the details of the graph now, but you can clearly see the blue shaded ones are the ones which are against killing the tumor cells. And these are zoledronate tumors. And then we also took fresh tumors and showed. So gamma delta T cells can mediate cytotoxicity, they can mediate ADCC. What is the relevance of this? Today, there are so many monoclonal antibodies in clinics. You have CD20, Herceptin, cetuximab. You can use a combination of gamma delta T cells along with these antibodies so that they can mediate ADCC. To prove, you have to do more experiments because you have to make your preclinical data more robust. So we did in vivo experiments and we showed that after giving an injection of zoledronate and gamma delta T cells, the last panel, you see there are a lot of apoptotic cells. So within 24 hours, you see a kill of the tumor cells in immunodeficient mice. We didn't remain, we showed the role of gamma deltas in solid tumors. We said we must give evidence to what happens to leukemias. And leukemias, mind you, are clonal malignancies, especially the T cell leukemias. So they are clonal arrangements, they are clonal populations of T cells. So the question which came to my mind is today there are standard protocols which are given to TALL patients, well-defined chemotherapy protocols. And these are, mind you, children, pediatric patients. And this is a very aggressive form of chemotherapy which is given to the children. nobody had looked at the clonal status of T cells. It was important from a clinician's point of view that the blast count goes down with therapy. The question I discussed with our clinicians was, should we find out how many of our acute leukemias are clonal alpha betas, that is alpha beta TCR, and how many are clonal gamma deltas? Well, they said, oh madam, you can, if you're interested, you can certainly do that. But beyond that, there was not much enthusiasm because they thought this is a research question. We did a whole lot of work in looking at gene rearrangements to establish clonality in these patients, and we showed that 30% of the acute lymphoblastic leukemia patients are clonal gamma deltas. So there is a difference. So obviously, the next question came from the clinician, so what? You know, okay, 30% of our leukemia patients are clonal gamma deltas, so what? So we did a follow-up and survival analysis. This was about 8 years follow-up doing clonality in these patients, and we showed that TCR gamma delta survive longer, while those with clonal TCR alpha beta succumb to the disease. So this is a beautiful marker. Upfront, if you look at clonality in these patients, if you know that they are clonal gamma deltas, they can be, you can spare them the chemotherapy. No need to give an aggressive form of chemotherapy. And mind you, these are again pediatric children. So this generated a lot of interest, and then we see one question leads to the other. Now we showed that there is a survival advantage to clonal gamma deltas. How does it offer survival advantage? So we did gene expression studies, and we traced down which are those genes which give the survival advantage. Then we came to the conclusion that the synaptic formation between gamma delta and the tumor cell is different compared to alpha betas. You know, because an effector cell has to come in close contact with the tumor cell and release the perforating granules at the site to kill the tumor cells. So you have to, you keep asking questions and provide evidence. So this was the story with gamma deltas. We showed that they work beautifully in solid tumors, they work in leukemias, but zoledronate also is known to have an action on the tumor cells directly. I mentioned to you the bisphosphonate. So we again did a whole lot of work in trying to understand what are the differential proteins, and for this we did a whole lot of proteomics to show, we used iTrack, and we showed that tumor cells treated with bisphosphonates, there are these changes which occur, there's downregulation of lipid metabolism, there's an EGFR downregulation, there's cell cycle arrest. Most importantly, we were able to show that they upregulate IPP synthesis catalyzing enzyme. See, in the beginning, I told you that there is inhibition of farnesyl pyrophosphate synthase, upregulation of IPP. So all this we could re-establish using a detailed proteomic approach. Fine. Next question was, patients who are given bisphosphonates, what happens there? Are there gamma deltas in those patients? What is the status of gamma deltas in this? I'm just showing you, each slide is like a whole project which was done. And so we did extensive immunoprofiling in breast cancer patients who were on long-term bisphosphonates. And we showed that these patients, I don't know if this works, we showed that these patients express CD25, they are activated, the middle panel in the upper figure, they are activated, they produce they express CD25 here, and they express the phenotype which is the TEMRA. TEMRA phenotype is the effector memory phenotype. So this tells us that in vivo gamma delta T cells are memory getting activated, they have the memory, so have the ability to kill tumor cells. So this was the evidence coming back from the lab data, going to the patients. Again, the research question which we wanted to ask was, what regulates the function of gamma deltas? How are gamma deltas regulated? Now, to ask this question, in the thymic environment, when there is a selection which happens and the lineage discrimination, that a particular T cell will become alpha beta or gamma delta, notch plays a very important role in lineage discrimination. So it was a wild thought in my mind, does notch regulate effector functions of gamma deltas? And I still remember when my student joined and I told Dinku that, Dinku, you are going to work on this project. He being a smart boy, he said, madam, there is no paper on this, that notch regulates effector functions. I said, that is the reason we are working on it. Why would we work if somebody has shown it already? He was not very convinced that time, but then slowly he got convinced, and he did beautiful work with me, and he showed that involvement of notch in activation and effector functions of gamma delta T cells. So a whole lot of siRNA targeting of the notch intracellular domain and showing that notch is the key molecule which regulates cytotoxic functions of gamma delta T cells. Now, having worked, as I said, one question leading to the other. So on the left-hand panel, you see that we had done a lot of work to show interactions of gamma delta T cells and tumor cells. But what is happening with the osteoclast? Because zoledronate, as I told you, also works on osteoclast. So that is when Swati Phalke joined my lab, and she had very good hands in cell assays. And it is very difficult to generate truly human osteoclast cultures. She did a lot of work on murine, which were easy to make, but relatively difficult to do with humans. And these multinucleated osteoclasts she could generate. And then we did a whole lot of work to do bone resorption assays. You see the pit assay, which is on the extreme left. You see pits, which are because of the bone getting osteoclast degrading the bone. And then when you give them supernatants from the gamma delta T cells, you see a closure of the pit. So this is a new angle to it, how understanding how bisphosphorates and gamma delta T cells work. We continue the story and we said, now let's look at epigenetic regulation of gamma delta T cells with checkpoint blockade using HDAC inhibitors. Now this was the project because then I applied for a Unit of Excellence project to DBT. It was a big project. It is done in collaboration with Sanjeev Belande, Dr. Sanjeev Belande from Phule, who is known for his epigenetic work, and Dr. Renti Banerjee making nanoparticles, and we all got together and we decided to tackle this project. You know that HDAC inhibitors are in clinics today. They are used in clinics, but there are a lot of problems with HDAC inhibitors. And all this while, people have been only studying tumor cells and what happens to tumor cells with HDAC inhibitors. We asked the question, what happens to immune cells with HDAC inhibitors? And to our surprise, we found that immune functions are compromised. You know, see the proliferation coming down with valproic acid or trichostatin or SAHA. We used all that and we showed that immune function is impaired. Of course, we did in CD4 cells, CD8 cells, and also gamma delta T cells. Even the cytotoxic functions coming down. Then we said, what is the reason? And then we showed that program death ligand 1, PDL1 and PD1. You know, today all the immunotherapy is directed against these two molecules, the co-stimulatory molecules, the PD1 and the PDL1. And they are upregulated after treatment with HDACs. And so naturally, if we block them, we showed that this could reverse. You block PD1, PDL1 on gamma delta T cells, and you can reverse the effect. So checkpoint blockade rescues the repressive effect of histone deacetylases inhibitors on gamma delta T cells. So having covered, and this project is still ongoing, and Dr. Sanjeev Belande is doing extensive ChIP-seq analysis on our gamma deltas treated with different combinations of antigens, and we are hoping for some good data to come out. There's totally a different story to it, and those of you who know the basic theory, the 3 E's of immunoediting, which was put forward by Hans Schreiber, you know that that the immune cells can become anti-tumor, and in the final stage can also take the shape of being pro-tumor and promote tumorigenesis. So this was the Jekyll and Hyde story of IL-17 producing gamma delta T cells, which we published first this review article. In fact, we were invited to write this review article because of the paper published in the International Journal of Cancer, which we published. And we said, now see, again, the selection, the choice is important. Now, where do I look at this inflammatory subtype, the IL-17 producing gamma delta T cells? These cells are the cells which cause inflammation. So I was thinking, why not look at gallbladder cancer? Now, this is one of the cancer which is rising in India, which is coming from the Ganges belt. And a lot of studies going on, and even at ADRAC, at our centers, the epidemiology group, totally devoted to working with, and there are some wonderful work on gallbladder cancers. So gallbladder cancers develop on the background of cholelithiasis, that is gallstones. So chronic insult to the gallbladder gives rise to inflammation and then to a malignancy. So I thought this would be a nice model to study in gallbladder cancers. So there's no data on profiling in gamma. So we did extensive immune profiling, and we showed that in the tumor infiltrates, and we have a subset which is delta, which are 17 producers. You know, So we devised a method to isolate. I'm telling you what I'm telling you in one line. It has been a backbreaking work. It's a very, very small subset to purify that from the tumor and from the peripheral blood is challenging. Rishikesh Patil in my group did this work. And we did extensive work on delta 17. We showed how they migrate to the tumor microenvironment, what are the chemokines, what axis the CXCL9, CXCR3 axis is used by them. So you do these dual chamber assays, you put tumor cells on at the bottom and the gamma delta cells on the top, and let the supernatants of gamma deltas go to the tumor cells. So beautiful assays, use antibodies to block it. So you can trace down what is the axis used by the gamma deltas to migrate to the tumor. We came across a very interesting observation that these cells produce VEGF, the delta 17, the vascular endothelial growth factor. It promotes angiogenesis. So we did the protein array for angiogenesis. We showed a whole lot of angiogenesis markers getting lit up after they are treated with the supernatants of gamma delta T cells, or with the delta 17 supernatants. So we did the classical CAM assays also, because you have to show a biological assay also. Even though you have done an angiogenesis array, you should support it with a biological assay system. And then we said, okay, the next question we asked, what happens to these patients? So we did again survival in these patients. And I must compliment the way records are maintained at the Tata Hospital. You can get it's possible to do such data. So we could look at those patients who have high delta 17, do not do well, those who have low do well. So this again becomes like a marker, upfront marker to look at, a prognostic marker. To look at overall survival. We looked at total IL-17, we looked at delta 17, we looked at interferon gamma, and so you can clearly see the difference, the reverse. If you have interferon gamma, better survival. If you have IL-17, the poor survival. Now I come to the area, when I took over as director at the Tata Memorial Center in 2013, and it's a beautifully located place. I think so many of you maybe visited, most of you have visited this place. And we have the Cancer Research Institute, we have the Jasawala Shodhika, which is the hospital, we have the Clinical Research Center, and we have Center for Cancer Epidemiology. And there are many, many more new buildings which have now come up. Nuclear medicine, we have bone marrow transplant unit, and so many more now which are coming up. There are different departments. Medical oncology, radiation oncology, surgical oncology. It's all surgeries take place here, and I absolutely manage with a team of efficient clinicians. And I was heading this center, both looking at the basic research wing, which had principal investigators, scientists, and also the clinicians. Of course, I had the support from the deputy director, who himself was a clinician, and the entire team was there with me. But I enjoyed doing all this. And it gave me a totally new perspective of looking at life. And I saw cancer patients from so close. See, being a scientist, you're at the bench. You're using samples, you're using surgically excised tumors, you're using tumors and lymph nodes, and everything comes to you on your bench. But when you look at people who are from where this has come, it gives you a shock. And again, I emphasize, you have to then start thinking that your research has to go outside your lab, and has to get the importance of getting it translated to patients. And I must tell you all, the young students, that you cannot work in silo. You cannot say, me and my lab and my papers. You have to collaborate. You have to throw your expertise open to all, and you have to invite others to participate in your project work. So I was fortunate enough to do this, and we worked on galactin-3 with Dr. Rajiv Kalraya, who was at the institute. And here I must also mention the expertise which we had developed in bone-related work. I collaborated with Dr. Madhavi Indab. She's a zoologist at the Rupali College, and she has these nice compounds coming from the mollusks, which she said can inhibit osteoclastogenesis. And she came to me and she said, and I've been an alumni of Rupali College, so I have naturally my feelings about the college. And so we did it, and we published this paper in Bone, which is one of the good journal. And then we had series of publications on not only this, on other extracts. And again, she sent her students to my lab. 3 of her students completed their PhD program with us, and it worked very well. Again, coming to collaborations and importance of the collaborations, there was a call from DBT, and DBT brought some of the scientists together and said, see, that those were the days when we're talking about the HPV vaccine, the human papilloma virus vaccine, because human papilloma virus causes cervix cancer. And the whole lot of argument going about vaccine, whether the vaccine is going to work in the Indian population, our HPV is different from the Western HPV, our this is different. But nobody had data. So we got together and we studied the molecular variants of HPV-16 associated cervical cancer in the Indian population. Several of the labs got together. We made SOPs, we did the RT-PCRs, we extensive sequencing, and we published this paper in International Journal of Cancer, and we showed how our HPVs, what are the differences, what are the variants in our HPVs. And we also looked region-wise distribution of high-risk human papilloma viruses, squamous cell carcinomas of the cervix in India. So this is all when I was doing all this with my main program, main focus of gamma delta T cells. Of course, you need funding, and we got generous funding for this. You could employ staff, and we could get the work done. Continue with the collaboration is here now. I collaborate with IIT, and the continuation goes on. The collaboration goes on. Prakriti Tanalia's lab. She makes these scaffolds, biodegradable scaffolds. So we're now trying to see if we can use these as a vehicle to deliver T cells to animals. A very nice this would be extension would be to the CAR T cell, the chimeric antigen receptor T cells now. Again, another collaboration with Dr. Dixit's group, where we studied the common genetic variation risk for gallbladder cancer in India, a genome-wide association studies, and he was my collaborator also on it's a part of the COE, the Center of Excellence for Gallbladder Cancer, which we got from DBT. So collaborations are absolutely important. You need to collaborate. And coming to industry academia, unfortunately, India, we do not have the collaboration the way it happens there. But there is an interesting story how we collaborated with Unilever. We were working on gamma delta T cells, and one of the ligands of gamma delta T cells are alkylamines. And alkylamines are found in grapes, in apples, in black tea. In black tea, it is ethylamine, which is one of the components, and thiamine, which can activate gamma delta T cells. So when one fine day I get a call from Unilever, and not Unilever India, it was from Unilever Netherlands. And there was a foreigner speaking, and he said, Dr. Chiplunkar, we read your work on gamma delta T cells. Would you be interested in collaborations? I said, fine. Then we had a lot of discussion meetings, and they said, we'll give you the black tea fractions, and you have to tell us whether they activate gamma delta T cells. I took a student on this, and that dedicated her to work on this, and we got some beautiful data showing that black tea fractions can activate gamma deltas, they can activate the cytotoxic function. So this is like a nutraceutical, you can say, which can help in activating gamma delta T cells. Then we used combinations of all herbs, ashwagandha and all, name it, which is used, and then looked at the modulation of gamma delta T cell function. In those days, there was an advertisement also coming up. Immunity makes the difference. There's an old lady shown drinking tea who's more healthier and happier. And so, it was all fun doing this work. Another project is still ongoing, and that's why I'm not sharing the data, but we did Bristol Myers School on evaluate the role of gamma delta T cells in colorectal cancer. Here again, they wanted to see the microsatellite instability and the gamma delta T cell infiltration into tumors at the peripheral lab. So this project just got over a few months ago, and we are analyzing the data. We talk of Ayurveda, and we always keep saying there is no experimental evidence and we should provide evidence. Then it is our responsibility to do that. And if you have the know-how, the expertise, why not extend it or why not use it to answer some basic questions? And I was very fortunate when there was a call initiated, Science Initiative in Ayurveda from Department of Science and Technology, Government of India, under the leadership of Professor M.S. Valyathan and Professor Chinnamani. I still remember that day when I got a letter from Dr. Valyathan saying that I would like to have your expertise as an immunologist to answer some of the questions in Ayurveda. He invited us to Bangalore, gave us a series of lectures. And I was very frank enough. I said, I don't understand anything in Ayurveda. He said, you attend the lectures, and then really it was wonderful how well read he is and well studied. Both of them spoke to us. And then we did the first project on immunological and it was a clinical trial, but immunological and metabolic responses, the therapeutic effect of basti in obesity. So we took obese individuals and gave them this basti therapy. And of course, this was done in collaboration with Dr. Urmila Thatte, who was leading the clinic, along with me, Urmila Thatte, and the Podar Medical College, Ayurvedic Medical College. And this study was published. Mind you, we had great difficulty publishing this. No American journal. We used to submit our papers to come back the next day. So it was impossible, because we had given the support and good support from the immunological side that we showed that inflammatory cytokines which cause insulin resistance are reduced after basti, after protocol of basti. We also did another project on understanding the immunomodulatory effect of Jasad Bhasma. This was with Dr. Bellari, who was leading this, and my lab looked at the immunomodulatory functions. So I feel I now want to appeal to younger students. Ayurveda is our ancient culture, and we must lend support, and we should give experimental evidence, evidence-based medicine, what I say, to this science. While doing all this, I had taken up a lot of responsibilities. I acted as a secretary president for the Indian Association of Cancer Research. I worked with the Indian Immunology Society as the vice president, Mumbai Immunology Group, which I'm currently the president, Immuno-Oncology Society, just a new society established 2 years ago. Our goal, and I was then invited to become the member of the International Union of Immunological Societies as the education committee member. All this is done to promote immunology as a subject, to promote cancer research, and we undertake a lot of programs. We did a lot of Karkarog Jagruti Abhiyans in schools, in colleges. Just picked up a few pictures just to show you all. But this is along with your routine work, you do and participate on a Saturday or a Sunday, go and talk about cancer research to students and teachers. It was all wonderful. Mumbai Immunology Group, I'm sure some of you have also attended our courses. We do hands-on training workshops, inflow cytometry or immunological techniques. And I must say I have a wonderful group with me. Dr. Jyoti Kode is there, Dr. Sinus is there. We all come together to give our time and efforts in organizing these workshops. Teaching and academic activities, Dr. Usha Mukundan knows. She has also been on several committees of the University of Mumbai, and I was a chairperson of Applied Biology Life Sciences for a very long time, and that is how our association is, grew, and we met more often, and several other committees of the university also. The Homi Bhabha National Institute, which is the deemed university where all the DA institutes now belong. So we have de-linked from Mumbai University, and since 2006, we are with Homi Bhabha National Institute. I was the convener of life sciences there. I was also Senate member for IIT Mumbai and mentored students. And this has been, of course, the most enriching experience to train PhD students and learn from them, teach them and learn from them. Finally, I'll come to the last slide. So many years, more than 35 years into cancer research and more focused on immunology. There is a dream left unfinished. I wanted to see during my tenure, to see that gamma delta T cell therapy goes to the patients. I wrote a project to the DAE. The project got approved. Money came in. The GMP facility is being done at ATRAC. Now, because of the COVID, it has got delayed, and I hope someday this facility will come up and using gamma delta T cells, because we have protocols for in vivo activation, in vitro activation, and finally also engineering and making affordable therapy. Because today the immunotherapy is costing $400,000. This is absolutely out of reach of anyone, I would say. So to make affordable, and there is DBT, ICMR, all these are funding now projects, and I'm on several committees. They're looking at funding projects for making CAR T cells or cell therapies. And this is one dream left unfinished, but I'm sure somebody will take it over, and some young scientists will come up with this form of therapy to be given to patients. Yeah. Finally, as heading of being a director of the RJC Center. As I said, you come in close contact with patients, the children. And when you look at these children, performing, moving about, smiling, you start wondering, what is it? What am I thinking? I'm thinking tomorrow to publish in this journal, that journal, get this project, get that project. And look at these people, how have they taken that life? It's full of energy, full of enthusiasm. Just they actively participate in programs which we organize. And as I told you, it has changed me a lot. I have discovered myself when I look at these. I'm not a clinician, I'm a scientist, but even then I love to interact with patients. There are some of them who are still in touch with me. During my tenure, a small effort from my side was to get funding for renovating the operation theatres and ICU at RJC, which I have been successful through generous donation from Morgan Stanley, because it involved a lot of dialogue with them, and then they coming inspecting, and it was not easy. Nobody gives money easily. So I enjoyed that too. And then coming out with one of the magazines for RJC called Andaram. And I thought patients, especially the bone marrow transplant patients who stay on the campus isolated for so long, this would be a nice event, to express their feelings. So we started this magazine, and I hope it continues. We have this issue come out just during the last year of my superannuation. We brought out this magazine. So as I said, overall, a very enjoyable journey. This journey has been long, and there are so many who helped me, who have helped me in this journey. And if I start taking individual names, I would need a lot of time. So all my teachers, and my teachers from school, from college, from Cancer Research Institute, from other institutes, students and trainees also, I'd say they are my teachers. Initially you teach them, and later they teach you so many things. Collaborators, all the clinicians, I have enjoyed working with them. Right from the beginning, I have collaborations with clinicians, scientists from Tata Memorial Center and from outside. All the funding support which I received from ICMR, PST, DBT, Indo-Journal project, Indo-EU project. The wonderful staff I have at Chiplunkar lab, they are like family members at RJC, all colleagues, friends, and of course, the most important, my family. I don't know, whenever I have a talk to give or something to do or to prepare, I just, I feel I ignore my duty as a family person, and they permit this very willingly. So I thank them. And this is our group, and some are there and some are not there, but some have gone out now. And the group now also, see, Vaibhav Chipre is there, so I took this picture. So this is nice smiling faces, and some of them have now gone abroad and settled, are doing postdoc, but the important part is they're still in touch with me.
Yeah.
And what I enjoy the most, and this gives me the most satisfaction. Thank you.
Thank you very much, and I think we'll also have questions.
If I've been able to see further, it's only because I stood on the shoulders of giants.
Thank you, thanks a lot. It was wonderful. And the way you narrated, I could see it's your baby because you've lived through all the projects. So we can take few questions, both from the participant, teachers and students. So you can unmute yourself or you can put it in the chat box. Yes, please. Yeah, please. Are there any questions, please? You can ask questions as to how, if you are really thinking of taking up research, or I'm very sure somebody, immunology is something which is very close to everyone's heart, right? And some of you are also taking some therapies. You could take, and if you could say something on autoimmune, what do you think?
You want me to say about what is autoimmunity?
No. If you could just say, like now we are hearing more. See, earlier, autoimmune diseases, like some of the diseases which earlier we had not even heard about it.
Right, see, because first the education happens in the thymus.
Okay.
The thymus is the center where your T cells learn what is self and what is non-self.
Okay.
It is there where something goes wrong. Because that's called break of tolerance. And this break of tolerance happens, and that's why autoimmunity is developed. It can be B cell autoimmune B cells, they can be autoimmune T cells. And these are against self-antigens, as I said, it's against your DNA, nuclear proteins, and there are ways in the general this give corticosteroids,
Yes,
steroids,
steroids.
suppress, dampen your immune response. It's a very complex issue, and there are so many things that comes with age. As advancing ages, it's found that there's an increase in incidence of autoimmunity. There are infections, viral infections, which give rise to autoimmunity. Because what happens, that's a question of molecular mimicry and cross-reactivity, that the antibodies and T cells, which are actually looking at the virus, start now looking at self-antigens because of the molecular mimicry. So it's a very complex and a very issue. There are psychosomatic reasons which can give rise to that. So there are multiple reasons, and
so you also believe you are what you think.
Huh?
Your immune system
Yes.
is tuned to your
very
thought
much.
process.
Your thought process plays a very important role
Okay.
in regulating your immunity. Yes, of course.
And has your lab any time worked on? Recently, there has been a lot of talk about this pigeon. Due to pigeon getting interstitial lung diseases.
Yeah, there have been articles to stop feeding the pigeons because of the COPD
droppings,
and yes, there are. We have not worked on it. We have not
really
data on that. Yeah.
Are there any questions? वो chat box में देखना. Yeah. Is there any question for madam? It need not be only with regards to cancer therapy or anything. It could be a general question also.
Students, you may ask.
Let's remove the internet connectivity. So if there are no questions, Captain Praveen Nayak,
Madam Ghorpade madam is there. She said she would.
She wants to ask the question?
No, she would propose a vote of thanks as a deputy.
Okay, sure.
And I would say that if there are questions, my email ID I have.
Yeah, sure.
Chiplunkar@gmail.com. I'm happy to answer your questions.
Yeah, Devashri is there?
Yes, Devashri madam.
Hello.
Hello, madam.
Yeah. Can you please put the vote of thanks?
Okay, madam. Yes, thank you. Yeah. Good afternoon, everybody. It's my privilege to propose vote of thanks on this auspicious day of 5th Savitribai Phule lecture series day, actually. I, Devashri Ghorpade, on behalf of Hindi Vidya Prachar Samiti's Ram Niranjan Junjunwala College and College Women Development Cell, first of all, extend my hearty vote of thanks to our distinguished chief guest for the day, Dr. Shubhada Chiplunkar madam, who spared her time for us today for Savitribai Phule lecture series. We had really an opportunity to hear thoughts of a hardcore researcher, scientist, and in the real sense, dynamic and iconic personality. Madam, your journey in the field of immunology is really inspiring for all of us, and your thoughts have really enlightened our minds today. Thank you, madam, for being with us. I'm also thankful to our director, Hindi Vidya Prachar Samiti, Dr. Usha Mukundan madam, because she has always been standing as a pillar of strength for all of us. I extend my sincere vote of thanks to our principal, Dr. Himanshu Davda sir, for his continuous support and guidance. A big thank you to all our vice principal for their continuous support. I would also like to thank the people from our technical team who have worked behind the scene to make this event happen, like basically technical arrangements and all these things. And last but not the least, I thank the wonderful students who have joined the talk in such a great numbers, without which this event would not have been possible. So thank you one and all, and have a nice day.
Yeah. Thank you, Devashri. There's a question from Tanya, madam. Madam will just reply to that, I think.
Yeah. Maybe the last that she's asked what.
Tanya, madam, what is that
you look at when you select students for your research? See, the students, when they come for to work with us for a PhD, they've already crossed the written exam, you know. So I know that they are good in their respective fields. Then is the interview, and that is the time, you know, when you're one-on-one looking at the student. I don't know. I feel I rely more on my own intuition and judgment, what I feel. And by that, I'm not saying that I would be impressed by the way a person talks. Or I am impressed when I just come to know that the student knows it. Maybe he's not able to express himself. Maybe there's a language barrier. That's fine. But I immediately get the hint that the person is bright, knows the answer, and I can see that dedication in that person. Now, you know, it's very difficult to say what are the measures, you know, I cannot pinpoint. But I'll tell you, I get that intuition and it has worked always in my case. So it's very difficult to answer your question. But yes, of course, your knowledge is judged. That's true. And then there is that X factor.
Spark.
That spark which comes and which is sensed. I don't know how.
Thank you. I think, Tanya, that was a good question because everyone wants to work with the best researcher. Yeah. Thank you.
Yeah. Thank you, ma'am.
Thank you very much. So, good day to all of you. DP sir, you can end the session. Thank you so much.
Thank you. Really enjoyed it.
Yeah.